Hello everyone, welcome to today's lecture on ESR or Erythrocyte Sedimentation Rate. ESR is a simple test that provides vital information about inflammation and other health conditions. In this lesson, we will cover what is ESR and its definition, the two methods used to measure ESR, which are Westergren and Wintrobe methods. Detailed step-by-step procedure for these methods, the types of ESR variations, normal values, factor affecting ESR, and finally its clinical significance. So let's get started. Now, what exactly is ESR? ESR or erythrocyte sedimentation rate measures how quickly red blood cells settle at the bottom of a test tube in 1 hour. When there is inflammation or abnormal proteins in the blood, the RBCs settle faster, increasing the ESR value. There are two main methods to measure ESR. The first is the Westergren method, which is highly sensitive and widely used. The second is the Wintrobe method, which is an alternative method often used when only a small blood sample is available. Let's move to the Westergren method, the most common and sensitive method for measuring ESR. Here is how it is done. First, we collect venous blood and mix it with 3.8% sodium citrate in a ratio of 4 parts blood to 1 part anticoagulant. This prevents the blood from clotting. Next, we fill a specialized Westergren tube up to the 0 mark. The tube is about 200 millimeters long. Then the tube is placed vertically in a rack, making sure it's undisturbed. The test is performed at room temperature, ideally between 18 and 25 degrees Celsius. Finally, after 1 hour, we measure the height of the clear plasma column at the top of the tube. This height in millimeters represents the ESR value. The Westergren method is highly sensitive and ideal for diagnosing inflammation related conditions. Now let's discuss the Wintrobe method, which is often used when smaller blood samples are needed. In this method, blood is collected using EDTA, an anticoagulant. The blood is then poured into a shorter Wintrobe tube, which is 110 millimeters long up to the 0 mark. Like the Westergren tube or Westergren method, the tube is placed vertically and left undisturbed for 1 hour. After this, we measure how far the RBCs have settled, which gives us the ESR value. This method is particularly useful in cases like pediatrics, where obtaining a large blood sample may not be feasible. However, it is less sensitive compared to Westergren method. ESR can vary due to both normal and abnormal conditions. Physiological variations include natural changes like higher ESR in elderly people, in females compared to males, and during the pregnancy. Pathological variations, on the other hand, occur due to diseases. ESR increases in conditions like infection, inflammation, autoimmune diseases, and cancers. It decreases in diseases like polycythemia or sickle cell anemia, where the blood becomes thicker. Let's look at the normal values of ESR. For the Westergren method, the normal range for men is 0 to 15 millimeters per hour. For women, it's 0 to 20 millimeters, and for children, it's 0 to 10 millimeters per hour. For Wintrobe method, normal range for men is 0 to 9 millimeters per hour. For women, it's 0 to 15 millimeters, and for children, it's 0 to 8 millimeters per hour. Several factors affect ESR. The shape and size of RBCs play a significant role. For example, abnormal shapes like sickle cells lower the ESR, while larger RBCs settle faster and increase the ESR. Plasma composition also matters. High levels of proteins like fibrinogen and globulins can increase the ESR. Finally, technical factors like the dimensions of the tube, type of anticoagulant used, and temperature can influence the results. Finally, let's talk about the significance of ESR. ESR is a key indicator of inflammation and helps in diagnosing diseases like rheumatoid arthritis, tuberculosis, and autoimmune conditions. It is also useful in monitoring the progression of diseases or the response to treatments, especially in cases like lupus or cancer. However, remember that ESR is a non specific marker. It should always be used in combination with other diagnostic tests for accurate diagnosis. Before we wrap up, let me share some fun facts about ESR that you might find interesting. Did you know ESR was first discovered in 1897 by Polish physician named Edmund Bernanke? It's one of the most oldest laboratory tests still used routinely in modern medicine. An extremely low ESR can sometimes indicate severe dehydration. So it's not just about inflammation. And interestingly, a high ESR does not always mean disease. It can also increase temporarily after a heavy meal or even during menstruation. Finally, ESR tests are often paired with CRP or C reactive protein tests for a more detailed picture in inflammation or about inflammation. I hope you found this lecture useful and engaging. If you did, please don't forget to like this video, share it with your friends, and subscribe to the channel for more educational content. Your support keeps us going. See you in the next video.